So I think we can start. So, bueno, buenos días a todos los que están conectados, ya sea por YouTube o Zoom. Y bienvenidos a este seminario del programa de Frontiers in Genomics, que es organizado por la licenciatura en Ciencias Genómicas, el Centro de Ciencias Genómicas y el Instituto de Biotecnología. Y como saben, las preguntas siempre son al final, ya sea que alcen la mano y les demos el micrófono para que puedan hacer la pregunta, o las pueden escribir en el chat. Y ya sea que Daniel lea la pregunta o yo la leería. Bueno, voy a pasar a la introducción. Para mí es un gran gusto presentar al doctor Daniel Geschwind, quien es Distinguished Gordon and Virginia McDonald Professor de Genética Humana, Neurobiología y Psiquiatría en la Universidad de California en Los Ángeles. También él es Senior Dean Asociado y Vicecanciller en Medicina de Precisión en el Instituto de Medicina de Precisión de la Universidad de California en Los Ángeles. El doctor Geschwind es MD PhD de la Universidad de Yale y completó su entrenamiento en neurobiología en UCLA, en donde ha desarrollado la mayor parte de su carrera. Daniel es pionero en transcriptomics y genómica funcional del sistema nervioso y su aplicación a métodos de biología de sistemas en enfermedades del cerebro humano. Su laboratorio ha demostrado que la coexpresión de genes muestra una configuración de redes que puede ser usada para entender mecanismos neuronales en salud y en enfermedad. A partir de esto, su grupo ha definido la patología molecular del autismo y otras enfermedades psiquiátricas. También ha hecho contribuciones muy importantes de nuevas rutas que participan en neurodegeneración y que pueden facilitar la regeneración después de una lesión. Ha tenido más de 70 estudiantes graduados y postdoc que tienen diferentes backgrounds de inicio de formación. El doctor Geschwind ha recibido múltiples reconocimientos y premios que incluyen, solamente para mencionar algunos, la medalla de oro de la Society of Biological Psychiatry, el premio Ron de la Brain Behavior Foundation, es miembro electo de la American Association of Physicians y de la Academia de Medicina de Estados Unidos, de la cual recientemente recibió el Sarnat International Prize in Mental Health por su trabajo seminal en genética de autismo. Y como les dije, tenemos el gusto de tenerlo aquí, aunque sea online, para hoy, y nos va a hablar sobre Defining and Refining the Molecular Pathology of Neuropsychiatric Disorders. Daniel, thank you very much to be here. So we think that we are honored that you can participate in the program, and we are ready to hear your talk.
Thank you very much. I'm really glad to be here, and I apologize. I understand the Spanish, but I don't speak Spanish that well, even though growing up in LA. All my children do, of course, but I didn't grow up here. So I apologize for for that, but it's great to hear the introduction in Spanish. I really appreciate it, and thank you for the generous introduction. So I'm going to talk with you a little bit, today about neuropsychiatric disorders and how I've been thinking about them. I'm trained as a neurologist, so I'm not a psychiatrist, but I've been very interested in any disorders of the brain, especially those that affect mental health. As you can see from A, the lifetime prevalence of these disorders is high. So anxiety disorders at the top, major depression between 15 and 20%, ADHD in the middle, PTSD also fairly high, ASD at around a little more than 1%, schizophrenia a little less than 1%, etc. These are common disorders. All of us have been affected by these disorders, either directly or indirectly by family members. And so, and part of the issue, and I'm not going to talk about that today, very much, is that these are part of the human condition. They're not something separate from it. I think it's the kind of flip side of our high cognitive and behavioral capacities. So we can think of these as disorders when they need treatment, but in some ways they're just part of human variability. The heritability of these disorders is all very high. And when you measure with genetic markers, common genetic markers, you also confirm that high heritability that you see from twin and family studies And again, autism is shown there. It's somewhere between 60 and 80%. And the SNPs don't capture all the heritability, so that's why it's a little bit lower. But what's really remarkable, so, there have been a lot of GWAS, a lot of genes identified now. And as we do genome-wide association and identify common genetic variants associated with these disorders, we can also take the SNP-based heritability and correlate it across these disorders. And we see that bipolar and schizophrenia are very high. That's perhaps not very surprising given the similarity of these disorders, the similarity of their onset, of age, and their tempo. Major depression and schizophrenia, though, have a high overlap, as does ASD and schizophrenia at 0.2. This is very significant. So what it means is that a significant portion, maybe a fifth of the genetic risk for autism overlaps with schizophrenia, but most of it is separate. But we have to deal with this. There are kind of shared and distinct aspects of these conditions. And so as we're studying them, we want to understand that. So just keep that in mind. So I'm going to focus primarily and start on autism. I'll introduce autism to you briefly. Autism is a disorder that involves challenges in social behavior primarily and repetitive restrictive behavior. And until the last DSM-5, language, but in DSM-5 they removed language. And I think that was actually wrong, but that's again for another day. But what we can see here and what I'm showing is that when there's a normal distribution, there's a continuum of the social behavior and social cognition and repetitive restrictive behavior, the flip side of which is mental flexibility. And so if you're in the red on these, you end up with a diagnosis of autism. About 30% of people with autism also have intellectual disability, and about 10% or so also have epilepsy. So there's a relationship between these neurodevelopmental disorders. And so my framing about these is that there aren't genes that are autism genes per se. Genes affect brain development, leading to susceptibility and increased risk. And the general idea of these is that a developmental insult during fetal life leads to abnormal circuit development and function that predisposes to autism and allied disorders. So what do we know about the genetic basis of autism? So what separates autism from the others is social cognition and mental flexibility, of course. The mental flexibility being the repetitive restrictive behavior, resistance to changes in routine, et cetera. And so this is a review from a few years ago, but I think it still stands fairly solid in terms of the main story here. And that is that non-additive, rare inherited de novo mutations, so the medical genetic conditions, recessive, dominant, de novo, like not Down syndrome, but like Down syndrome. So they're genetic, but not inherited, account for around 10%. Then common inherited, about half, and then unaccounted for, about 40%. And if I were giving a straight ahead genetics talk, I would be telling you about our discoveries of rare inherited variation that's also accounting for some of that, as well as there's probably room for discovery of more rare mutations. So the notion is that autism, there's about 10% of it all the way on the left that's a collection of rare disorders that's known, and probably room for another 5 or 10% more. So up to 20% is likely to be due to these rare disorders, and the rest a combination of rare and common inherited genetic variation. And what we've learned from this is that we can do clinical testing and identify a mutation in about 10 or 15% of cases, that there is not one autism gene, there are many genes. In fact, there have been hundreds now identified at high confidence that are rare de novo mutations. None account for more than 1% of cases. Perhaps Fragile X is the most common, around 1% in most. Here we have it around 2%. It varies depending on ascertainment. And there's strong pleiotropy. In other words, very few of these mutations are specifically causing autism. They cause usually broad neurodevelopmental syndromes that involve autism. or they can cause intellectual disability, and sometimes even a kind of childhood onset or or schizophrenia. So the challenge to the classic genetics paradigm is there's genetic heterogeneity at the population and individual levels. We know that genes don't act in isolation. They're components of biological signaling pathways. And in the brain, more so, even complex neural networks. So one of our general thoughts has been, can we identify what I'll call convergent or shared patterns that are related to key aspects of biological function or disease pathophysiology? In other words, are these genes impacting shared pathways? Are there phenotypes that are part of shared biological function? And can we use these patterns to inform treatment development? And the way we've looked at this is using what I'll call a systems biology approach. That is, you start with obviously having the phenotype data and the phenotype, but you dig deep in terms of identifying the genetic causes from sequence or copy number, epigenetic alterations, gene expression, et cetera. And so from a kind of model standpoint, the notion is genetic variation percolates during development to RNA, to protein, to cells, to in brain, to synapses, the things that are connecting the neurons, creating circuits that then lead to behavior. From the standpoint of the genome, we're talking about 3 billion base pairs and leading to around 300,000 genes, and measuring at the RNA level, depending on whether you're looking at non-coding, coding, et cetera, it can be quite a bit more. The point is not that mRNA is the most important thing in the world, but that mRNA is measurable inexpensively in very high throughput, now even at a single cell level. So mRNA has been a tractable intermediate for querying what the relationship between DNA and phenotypes. So there are two ways that we think about that. One is in the forward genetic way, obviously, where you have disease A variant, this is just a cartoon, or variants, and you ask kind of how they lead to the phenotype, measuring RNA, which is, again, a quantitative molecular phenotype that you can measure in high throughput at reasonable cost that has high dynamic range as well. I can measure from 1 copy to cell to 100,000. A kind of reverse genetic way of thinking is, let's measure the omics, right? And then kind of try to understand how it kind of connects to genetic risk. And so that involves studying the brain. And so I'm going to talk today primarily about our work studying the postmortem brain of autism. Here's the key framing idea. Neurologic disorders like Alzheimer's, Parkinson's disease, ALS, have visible macroscopic and microscopic pathology. But psychiatric disorders are considered differently because of their lack of defining pathology. In fact, that's almost a definition of a psychiatric disorder. But we all know, hundreds of years ago, it was fine to say that psychiatric disorders came out of some spirit or something. But now we know that they come from the organ of the brain. They come from our physiology. So we reasoned that there had to be defining pathology and that they just weren't visible at the microscopic or macroscopic level. So we used genomic methods to look beyond microscopic resolution to identify and define a molecular pathology in postmortem brain from subjects who had autism. And what really struck me in our first study, which was led by Irina Voinigo, who was a postdoc in the lab, who's now a professor in Australia at the University of New South Wales. And she's gone on to do beautiful work in genomics of nervous system diseases. But this first study, we only had postmortem brain from frontal temporal lobe and cerebellum of about 19 cases and I think 20 controls. Now, we studied the frontal and temporal lobe because those are regions that are involved in social cognition and that had been implicated by imaging and other behavioral studies. We found a pattern that was very strong in frontal and temporal lobe and fairly absent in the cerebellum. That is, downregulation of genes related to synaptic vesicle signaling, some GABAergic markers, and upregulation of microglia and astrocyte genes. In a subsequent study, we saw that we were able to refine this substantially in that the downregulated genes fell into neurons, but broke into 3 groupings of downregulated genes representing different fundamental aspects. I'll get to how we did this in a second. And then the neuroinflammatory group broke into an astrocyte and microglia group. And this was published in 2016, 5 years after the original paper, and also included about 3 times as many subjects. One of the other things that we found, and this is just showing those 2 cell types, was that, so at that point in 2000, our major finding was we had this characteristic, let's look at number 2 first, molecular dysregulation, increase of neural immune and inflammatory genes, microglia and astrocytes, and decrease of very specific synaptic gene expression, not all neuronal genes. It was synaptic vesicle recycling, bulk endocytosis, and some local circuit inhibitory neuron genes. But another thing that Irina saw that was really remarkable is when we compared in controls, the 2 regions, the frontal lobe, BA9, to the temporal lobe, BA41, that's Brian's area, we got about 500 genes differentially expressed. However, when we looked in autism, it was 1/10. So we saw this as an attenuation of the normal molecular differences between cortical regions. And this was pretty remarkable. We had seen this initially in the Vonnegut study, but we were able to validate it in this much larger study. And again, genes don't act alone. So showing genes that are up or down doesn't show everything. And I've kind of alluded to that in some of the figures before, but I haven't explained why. So here are 2 genes, gene 1 and gene 2. You can see in these box plots that neither of these genes are differentially expressed, even at a nominal level. However, when you look in the control group, these genes co-vary. Their expression co-varies substantially over the samples, the brain samples that are measured, whereas in the experimental group, this covariation pattern is lost. And so we reasoned very, very early on with collaboration from Steve Horvath and his laboratory that actually it's not just genes going up or down, it's how genes co-vary that's actually very important. And so we developed, and so it's how genes are connected. And so the notion is if 2 genes like this co-vary substantially, we can actually create a network where the edges between genes represent the extent of this covariation. The stronger the covariation, the stronger the correlation, the stronger the edge. So in all of our work, network analysis provides an organizing framework beyond just differential expression. And there are a lot of reasons for that. One of them is that, if I have 500 genes that are different between autism and control, or 2,000 or 3,000, how do I know which are important? How do I know what biological functions? The network analysis groups genes by their covariation. So that here, we're again in this cartoon, expression level versus sample. And you can see that the genes are grouping into 3 groups based on this covariation pattern, this correlation, from which we create groupings, clusters that we call modules. And within modules, we have central genes called hub genes that are kind of the main identity of that module and really represent its eigengene or eigenvector. Now, once we identify a module, we can then talk about it and try to understand, investigate it. So we can ask, is there an enrichment in certain kind of genes here? Do they represent certain cell types? You can imagine that across 100 different brain samples, that a major source of covariation is going to be the contribution and the variability in cell type composition, slight differences in cell type composition, either due to dissection artifacts or differences in people, will create covariation. And so one of the things that these modules represent are actually cell types. When we take bulk tissue and run this network analysis, we find that the modules represent one of the major sources of variation, which is cell type or synapses or even organelles, et cetera. So they represent, when genes covary, we infer that they're co-regulated, although we have to show that. And that co-regulation almost always corresponds to some fundamental biology that's linking those genes together. In many cases, that's because they're co-expressed in specific cell types across tissues, but it may be that they're involved in the same biological function, like mitochondrial function, RNA processing, et cetera. Activity-dependent gene regulation in the brain. So we've shown over the years that the brain network structure is robust and reproducible across regions, that its network position of a gene is biologically meaningful, that groups of these co-expressed genes called modules correspond to key elements of biological function. And within these modules, we can identify the hub genes. And this structure serves as a basis for identification of biologically meaningful insights. If you have a module that is no longer preserved in a disease, you now know that there's a change in that biological process. It's better than looking at one gene at a time. So you ask, what modules or hubs are different in disease and controls? So getting back to our gene expression studies from the Parikhchak et al. Nature, we had these neuronal modules, M4, M10, and M16, which represented different aspects. And I think this is showing M4 here. And we can see that this neuronal module is enriched in autism risk genes. What does that mean? Think about it this way. When I'm measuring gene expression in the brain, I don't know whether it's a cause or a consequence of having the disorder. That's why many people prefer the forward genetic approach, because you can easily infer causality. Here we don't know. Could this be because the person had autism or not? Well, there are ways to begin to look at that. You could look at, how do these relate to the length, somebody's age? If somebody's had autism longer and the module is more down-regulated in their brain, maybe you could say this is due to having autism. But really, we think the best way to get a causal anchor is to ask, where do risk genes sit? And in this case, we find that the risk genes sit in the downregulated neuronal modules, not in the upregulated glial modules. So we begin to have a causal model. What is ASD? So let me put it into a biomedical context. Cancer in general is a disorder, a collection of disorders, of course, of proliferation and invasion. All cancers share proliferation, abnormal cell proliferation, caused by a combination of genetic susceptibilities and, in some cases, environmental contributions. Asthma is a disease of hyperreactive airways due to environmental challenge in people with certain backgrounds. Stroke is a disorder of acute brain hypooxygenation due to many causes. It could be cardiac, it could be vascular, etc. So I'm trying to find the core cause model of the mechanism of disease. So our working model of ASD is not as elegant yet, because we don't really have even a model of how the brain works normally. But here's our working model based on our, at that point in 2016. We have causal genetic variation that sits in this ASD-M12. So there's altered neuronal gene expression due to genetic perturbations likely happening during development that leads to synaptic dysfunction and synaptic dyshomeostasis, which is propagated by astrocyte and microglial neuroinflammation. This is not inflammation like peripheral inflammation, like getting an infection. We call it inflammation because these cells are related to immune cells, and the genes are. But what it really is a defect or alterations in synaptic homeostasis. So we see increased microglia and astrocyte gene expression, and this is what characterizes ASD pathophysiology. So now that we've shown over a period of 5 years, going from 20 samples to 113, that this is a reproducible pattern, that this is somewhat generalizable, and it's seen in about 2/3, about 70% of cases, so not in every case. We think it's associated with severity, but we really don't know yet. But one question we can ask is, given the genetic overlap that I showed you in the second slide, is this transcriptomic pattern that we're seeing specific to autism, or is it generalizable? How is it manifest across disorders? So we took data. This is led by Mike Gandel when he was a postdoc in the lab, with a lot of help from Jill Haney, who was a graduate student, and Neil Parikshak. But Mike really drove this study, really remarkable study. Now Mike is an associate professor at University of Pennsylvania. So what we essentially found is that in autism, again, we see, and this is downloading hundreds and hundreds of microarray data from the web, from data sources. We spent a year normalizing them and making sure that everything is integrated properly. This was a huge bioinformatic project. So we could see that neurons and the synaptic genes were downregulated in autism, to a lesser extent in schizophrenia. There was a sharing of activity-dependent genes that were downregulated, and kind of plus or minus in bipolar. We saw increased astrocytes in all 3 disorders, in fact, more in schizophrenia than in autism. But the microglial upregulation was specific to autism. So one of the questions here is, again, are these just postmortem artifacts? Well, you know, we had other samples like from depression and alcoholism, which did not show these changes. So we had some evidence that this is not just a general thing. These are quite specific. But what we were able to do is ask, if we look at the correlation between the transcriptomes in these disorders across them, using the published microarray data and new RNA-seq data that we had generated as part of the PsycENCODE consortium with Kevin White's lab, we saw that the pairwise SNP correlation between disorders was highly correlated with the transcriptome correlation, suggesting that almost 50% of the variance in transcriptome overlap is related to shared common genetic variation. That's telling us there's a huge genetic component here, likely. We've gone on to really refine these patterns remarkably in the PsycENCODE consortium. And there's so much in these papers that it's hard for me to, I could give an entire talk just on one of these papers, but it would be kind of potentially only of interest to the aficionados and maybe too detailed. But in PsycENCODE, what we've been trying to do, just like ENCODE, is define the regulatory architecture of brain from embryonic fetal stages through development and across multiple brain regions, as well as how that is perturbed in disease. So we can use that to kind of connect this molecular pathology to genetic susceptibility. And so in this particular transcriptomic study, again led by Mike Gandel and published in 2018, we compared autism, schizophrenia, bipolar, and controls, nearly 2,000 samples. We had genotypes and RNA-seq. We were able to analyze both the gene expression as well as transcript isoform expression to look at local splicing. We did, of course, our network level integration, and we found several very interesting things. First, massively refined networks with this large sample size. We're able to really parse things very nicely. We even found a drug module just seen in patients getting antipsychotic drugs, bipolar and schizophrenia, not in autism. We identified specific neural immune pathways and trajectories. kind of, this is cartooned at the bottom here in this biological insight panel, where we can see that the microglia and astrocyte responses we're able to see, as well as shared across multiple immune cells, interferon response containing the majority of the known interferon response genes, as well as NF-kappa B pathways. And we can see across these disorders, they have quite different trajectories. Not surprisingly, the autism peaking much earlier than the bipolar and schizophrenia, coincident with the trajectory of onset of these disorders, autism being a childhood disorder and the others being adolescent or adult onset. Another thing that was really remarkable that Mike identified is that gene expression changes were broad, but isoform dysregulation was even more profound. That is, specific isoforms gave much more disease and cell type specificity. So it became clear that splicing and its regulation and understanding isoforms is going to be critical, which is now available with long read sequencing. And we started to do this in collaboration with Mike. So here's our refined disease model after years of study, after literally a decade. Kind of remarkable, I think, is that we have rare de novo variation and common genetic variation. I didn't show you all the data, both for schizophrenia and autism, sitting in synaptic dysfunction. There's also this kind of neural immune response involving endothelial cells and parasites, which make up the blood-brain barrier. So there's some changes in the blood-brain barrier. We don't know if it's disrupted or not, but we call it disruption. We have astrocyte and microglia upregulation as well. So autism involves all of these. Schizophrenia really involves primarily the synaptic dysfunction and to some extent astrocyte without the profound microglia upregulation chronically seen in autism. Bipolar is a much subtler condition. So this is kind of amazing. I just want to stop and reflect that these disorders that, 100 years ago weren't even thought to be due to brain. We can now define a brain molecular pathology using modern genomic methods and begin to use it in a very gross way. Kind of, again, the analogy is similar to cancer and proliferation and invasion. So we've now gone to ask, how broad is this pattern? Is this just in the frontal and temporal lobes, these regions of the brain that are involved in social cognition, or is it a broad cerebral cortical dysfunction? So Mike Gandol has led a study with Jill Haney, who was a graduate student that Mike and I co-supervised, as well as Bree Walmsley, who's a postdoc in the laboratory. 808 samples across 112 subjects, broken up as shown on the left, mostly male. And this is unpublished work, by the way. So this is showing, basically, if you look here, that this is the gene and that's transcript. Again, we get a much stronger signal from transcripts, as we mentioned. That if we look whole cortex, if we pool all the regions and do all regions in cases versus controls, we get almost 10,000 transcripts differentially expressed out of about 50,000 identified. We get about 4,000 genes out of the 30,000 dysregulated. And it varies across the region. And it's not just due to power, because you can see the sample sizes are relatively consistent. In fact, BA9, which has the most samples, does not have the most differential expression. In fact, or Brodmann's area, 17, which is the visual cortex, has the most differential expression. And we can look at this across the brain. And this is just shown here in this kind of cartoon where the frontal regions are yellow, parietal regions are this orangey, green is the temporal, and BA17, which is really distinct, which is the occipital cortex, visual cortex, is purple. And you can see that the effect sizes are very consistent across regions, but slightly greater in the occipital cortex shown here in purple. So if we look, we see this kind of brain-wide pattern, but with really striking and unexpected major changes in occipital cortex. Wow. That was really striking. And so this, surprising changes in sensory motor region, not primary association, but in BA3125 and BA7. Okay, that's pretty interesting. We'll get back to that. Why do we see this striking posterior, anterior to posterior gradient? Well, another thing we see that's related to that is a posterior attenuation in cortical patterning. Remember, I showed you that the differences between temporal and frontal lobe were markedly attenuated, almost absent in autism. And this is showing a cartoon of the difference between, comparing Brodmann's area 17 to BA 41, 42, 22, which is temporal lobe. And we can then permute the label, whether you're control or autism. And so we do a permutation to ask, is the difference that we see in controls different than cases? And is that difference more than expected by chance? And what's shown here, and the red lines show these comparisons, and the negative log 10 p-value, these are very significant differences, primarily between visual cortex. And so what we find here is that if we look at the first principal component of differential expression, this is differentially expressed genes, we can see that the downregulated gene, we see that these genes that are typically expressed in neurons that are downregulated are actually not showing the usual gradient in cortex. Usually in controls, you can see in green, there's an anterior to posterior gradient of increasing expression of these neuronal synaptic genes, but that's attenuated here. And vice versa, we see the same thing with the astrocytes and oligodendrocytes, the glia, that there's an attenuation of the normal. Normally, these go down from anterior to posterior, but they kind of stay pretty similar. Question is, why is this? And we'll get to that in a bit. Because really, to get to that, we have to ask some fundamental questions about cell type composition of these regions. One of the things that we know from studies in primate, but not in human, it actually had not been done before this study, is that there's an increase in neuronal density and an expansion of layer 3-4 from anterior to posterior in primates. So one of the things we reasoned is that this had to do with the expansion and the increase in neuronal density, making this a easier to detect phenomena in those regions that have higher neuronal density and higher packing. That was just a hypothesis. So let's stick with that. So now, do we find some of the same things that we found before in terms of the relationship of genes that are causal? And indeed, I'm just showing you one module here. There's this module, and the blue is showing the cortex-wide downregulation. Again, high in temporal lobe and parietal lobe and occipital lobe. This is a module called Gene M5 who's enriched for genes involved in synaptic transmission, neuronal projection, morphogenesis, cognition, synaptic plasticity. And it's significantly enriched in rare and common genetic variation. Again, similar to what we've seen before. This is showing that this is a cortex-wide phenomenon. It's not specific to any region. And again, it's just a replication of our previous findings that the genetic risk sits in the neuronal population. So then we did single cell sequencing in 3 regions: frontal cortex, parietal, and occipital, just from 6 subjects of autism and 6 of controls. We didn't, it's a very expensive study. And we felt that that would be enough just to ask, are these changes that we're seeing cortex-wide, downregulation of neuronal genes, upregulation of glia, blah, 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 is that all due to just changes in cell composition, or are these changes that are occurring within cells and cellular pathways? And so this is just showing our UMAP plot, showing that the cases and controls largely map onto each other, but it looks like there might be more of certain cells like astrocytes, microglia and autism, but pretty well map onto each other. And when we look at it, we see a very minimal change in cell type proportion. Again, remember, we only have an N of 6, and now we've expanded this to N of 30. And we do see very small, subtle changes in cell type proportion here. You can see in the autism, that, there might be, some changes, but they're really not significant. You can see the interneurons and controls in autism don't really differ. In autism, the excitatory neurons look like they're a little bit higher, in the posterior regions, more density, so to speak, but it's not that significant. But when we look at cell type specific gene expression, we see massive changes within thousands of genes, even in the small cohort, significant in FDR of 0.05, in excitatory neurons, in inhibitory neurons, as well as in the astrocytes, microglia, parasites, endothelial, et cetera. And previously, we've had no signal in oligodendrocytes, really, but looking at the single cell data, we're beginning to see that as well. And again, if you look at the purple, the most profound changes are in occipital lobe for the vast majority of these, and then frontal, just like we see in bulk tissue. So this is telling us that gradient that we're seeing is due to changes that are occurring within cell types and not due to cell type proportion, which is really an interesting head scratcher. So how does this brain-wide, kind of cortex-wide dysfunction fit with other data that we have? I'm just going to very briefly, because we're beginning to run towards the end, show you some additional data. Chris Hartle, who was a graduate student in the lab, who finished in 2020, I think, took the GTEx data, because one of the things we wanted to do is understand how modules and cortex and different brain regions all fit together. Nobody has actually done that. Usually people will study a few tissues at a time, identify modules and say, oh, this is the cortex module, but it's not really known. So GTEx profiled a lot of different tissues, and you can look here from cerebellar and cerebral hemisphere, prefrontal cortex, amygdala, hippocampus, hypothalamus, so cortical and subcortical regions. And so what we did is we created networks in each of these tissues, because they have 50 to 100 individuals. And so we can create networks. And then we used essentially something like a Jacquard index to kind of merge modules and see which ones were the same. So we could identify brain-wide modules that were represented across multiple regions. We could identify regional modules, let's say those that were just seen in the cortex, or region-specific modules. And then we did a lot of study with it. And I urge you to look at this paper if you're interested in models of genetic disease, because we actually tested the omnigenic model and it really doesn't fit. The disease really fits a polygenic model. But anyway, there's a lot of interesting stuff in this paper, but I'm just going to focus on one thing, which is that here we find that both for schizophrenia and autism, that most mutations sit not in specific regional modules, but again, in modules that are represented across many regions. So that means the impact of mutations is expected to act across multiple brain regions, cortex-wide, just as we see in the profiling study that I just showed you, that's still unpublished, but in press now. We see the same, this is with de novo loss of function mutations. And one of the points I want to make is that these neuronal modules indeed overlap with what's going on in the autism brain, in that there's an activity-dependent gene regulation endocytosis module called cortex M3 that is actually disrupted in the autism postmortem brain. It doesn't, it's not nearly as connected. So again, this fits with what we've seen before. And this module is very similar to the brain-wide M4, but it's cortex specific because it's involving activity-dependent gene regulation. This is another module that's brain-wide, and interestingly, its gene ontology annotations include social behavior, disrupted in autism, of course. And then another brain-wide module, which is involved in neural differentiation, migration, morphogenesis. This contains highly constrained genes that overlap with a module that we identified, two modules that Neil Parikshack identified a decade ago that were enriched in autism genes, but that are expressed in fetal development. So this is a persistent module that includes genes involved in neurogenesis, and there are stem cells in the ventricular zone still in adults that kind of persists in autism. And then the same thing with GWAS. We're running short, so I'm just going to go to some of the end. So the summary of this part of the talk, and I'm going to show you some single cell data and then end, is that the transcriptome is a quantitative molecular phenotype for forward and reverse genetic studies, that autism, schizophrenia, and bipolar share molecular pathology, but it's much more profound in autism, and that there's a brain-wide but posterior gradient emphasized dysfunction in autism that impacts primarily these layer 3-4 excitatory neurons, which have expanded posteriorly. So now I mentioned to you, again, more unpublished data. We've been doing a very large-scale RNA-seq study just in frontal cortex. And we did RNA-seq and single nucleus attack-seq. We did composition analysis, cell type differential gene expression, and we identified the regulatory networks that are driving differential gene expression. And we confirmed some of the laminar patterns with spatial transcriptomics. So what I'm just going to zip through this quickly. It's unpublished. This is just showing differential gene expression. And I just want to bring all of these neurons. These are excitatory neurons, superficial, middle, and deep, all enriched for ASD risk genes, enriched in neurons. Especially layer 2-3 excitatory neurons, which are the neurons that connect the two hemispheres and connect regions of the cerebral cortex. What's also interesting is that the layer 5 neurons down here that have differential expression, the layer 6 and 5, right here, 2 and 4, that are also downregulated, those also are the callosally projecting neurons, like the layer 2-3 neurons. They connect brain regions. We also see dysfunction of somatostatin neurons. We see an increase in reactive microglia. So what we see is not a huge change in the numbers of microglia, but a total change in their state from homeostatic to microglia. And the same thing with reactive astrocytes. That's why we didn't detect in the bulk tissue much of a change in cell type, because there's actually a change in cell state that's quite profound. I'm gonna skip through this because of time, but the point is we've been able to really annotate these genes in gory detail to see both pre and postsynaptic changes. And again, the study with spatial transcriptomics using nanostring, not super high resolution, but we're able to see layers, shows a kind of profound superficial layer 2-3, which starts tapering off in 4 and tapers off in 5-6. Now, we see the same thing with the interneurons, which is really interesting. The interneurons don't have laminar specificity. In other words, we can't find a layer 1 interneuron, a layer 2 one. But this is showing that the majority of the differential expression is occurring in the superficial layers. And we'll get back to that. We see the same thing with the glial cell. So we're seeing a profound superficial, primarily dysregulation, but not entirely superficial, but it's more so. So we wanted to now identify the regulatory drivers, right? Like we have all this differential expression, and we can use modern methods like scenic to which allow us, they builds co-expression networks and then prunes the networks into regulons based on transcription factor co-expression relationships. And so we score the regulons, and then so we can identify disease and cell type specific regulons in certain. So this is the cell types by diagnosis and the regulons. A regulon is transcription factor and its downstream genes. And we can see that we have the layer 2-3 down regulons, the astro up regulons, the microglia. So now we know that these are coherent biological processes that are being driven by known transcription factors. So that even, further emphasizes the coherence of the biology that we're seeing. What's remarkable again about the layer 2-3 down regulons is that 22 of 70 of the regulons enriched in layer 2, 3 that are down are high confidence safari genes, which are mostly de novo or Mendelian genes shown here. It's highly significant. So again, we're finding that the signal for genetic risk sits in neurons, and here specifically layer 2, 3 neurons. So to sum up, we have changes in cell composition. We have cell-specific ASD disrupted transcriptional signatures in these cell type regulatory mechanisms. So the ASD cortex is impacted by genetically driven neuronal dysfunction and a shift from homeostatic to reactive glial states that converges on the more superficial layers of cortex, but also involves other circuits as well. But that's where it's super profound. So I've shown you this kind of reverse, most of what I've been talking about is this reverse genetic omics quantitative stuff. What do we know from forward genetics? And again, I alluded to this by citing the Parikshack paper. But in 2013, we actually began to look at this to ask when and where do autism genes, what networks in normal brain development do they impact? And just to summarize this paper, which is published, they impact early cortical development, affecting transcriptional regulation and synaptic development, but again, primarily in the superficial cortical layers from layer 2 to 4. And in fact, more recent work that we've done in making fetal brain networks that was published just 3 years ago, it's not only rare genetic risk, it's common genetic risk. So if we ask where autism heritability is actually enriched in co-expression modules in normal fetal brain, we see this huge increase in this yellow module, but not the other modules. And in fact, this yellow module is fetal upper neurons. Again, confirming the work done almost 10 years ago, but now having, instead of a handful of autism risk genes like we had in 2012, 2013, now with hundreds. And in fact, this yellow module overlaps with that previous module identified very, very significantly, an odds ratio of 7. And in fact, if we look for the safari risk genes from the Simons Foundation, those with the highest confidence, they're massively enriched in this set of cells in this network. And that's just shown here by a postdoc in the lab, Katie Eyring. So in conclusion, human genetic studies are transforming our understanding of psychiatric disease etiology. Integrative genomic and gene network approaches provide organizing principles for understanding molecular phenotypes in the brain, defining points of potential biological convergence in disease. Genetic risk overlaps with the drivers of transcriptomic changes in brain, which impacts neurons involved in cortical-cortical connectivity in superficial layers of the cortex, as well as, of course, local circuits that are being impacted, and glia, which we presume are secondary. These genomic studies provide important mechanistic clues and a foundation for understanding how genetic risk propagates across development from these early neuronal modules all the way to impacting the deep layer of superficial layers of the brain. So again, a lot of people, I think I've mentioned them as we've gone along, but Bree Walmsley, a postdoc in the lab, has been leading all the single cell work with some help from Natalie Hawken and Jennifer Grunman and Michael Margolis, all grad students in the lab. I mentioned the work of Mike Gandel, of course, and Jill Haney in the cross cortex study, again, that's in press but unpublished. Again, others in the lab who have contributed over the years. My collaborator, Steve Horvath, of course, who used to be at UCLA, who moved to this aging company, and Giovanni Coppola and Bogdan Pasanuik. Again, Giovanni's now at Regeneron Pharmaceuticals. I'm indebted to NIH for developing the PsycEncode project, which underlies a lot of our work, as well as the Safari, the Simons Foundation, and Paul G. Allen Frontiers. Thank you.
Okay. Thank you very much, Daniel. So very interesting talk. So now it's open for questions. So I called you, you can raise the hand, and I can see someone raise the hand for a question, or you can put the question in the chat. There is one question. Luis Covarrubias, please.
Hi, thank you for your talk. I just want to ask you about the developmental origin of psychiatric diseases. And regarding this, I have just a particular example that I don't know if you are aware, but because it's an old-fashioned probably paper, it's a recent paper, but it's done apparently by just a correlation. And they found that the haploinsufficiency in DLL1, this notch ligand, cause different psychiatric diseases, including autism. They say they have intellectual disability, autism, and seizures. And we have a mouse line also with DLL haploinsufficiency, and they have a variety of neurological things. I don't know how to call them. There are symptoms of that. They have just a neurological deficiency. I just want to ask you regarding the notch pathway. Among your data, have you found a specific developmental pathways that are associated with autism or a particular syndrome?
So it's very interesting that you bring up, I mean, another talk that I sometimes give is titled The Neurodevelopmental Origins of Psychiatric Disorders. And so there's really, it's not surprising that autism has a neurodevelopmental origin. It almost has to. But when you look even at things that are adult onset, they have enormous, the genetic susceptibility for even adult onset psychiatric disorders is primarily neurodevelopmental. In schizophrenia, it's about 50-50 adult, kind of postnatal versus prenatal. But in major depression and others, it's strong. So if I understand your question properly, I'm strongly in favor of that. In fact, I've been writing for years a short review about this issue. I think it's a profound issue. So thank you for bringing it up.
And did you find a notch pathway? I mean, the notch pathway within, I mean, if you arrange the genes or the transcriptional data, did you find a notch pathway that's a unique pathway, or is just one among many others?
Yeah, no, it's one among many others. The Wind pathway, there are a bunch of neurodevelopmental pathways that are impacted. When you look at the transcription factors, you'll see, a whole bunch.
Okay, thank you very much.
So there are some questions in the chat. I don't know, Daniel, if you can read it, or do you want me
to
read it?
I can read it.
Okay.
Have you done network topologic analysis? Well, I'm not sure. That's from Fabel. I'm not sure what you're referring to. The analysis that we do, I didn't go into it in detail at all, none of the methods. But it's fundamentally, I use the term correlation, but because that's the foundation, but all of the analysis is based on topologic overlap. So it is a topological network analysis. I'm just not sure what you're, in fact, all of these are based on kind of graph theoretical principles and topology. I'm just wondering what you're asking, and feel free to ask the question if I haven't answered it, but conserved or enriched patterns. And so that's exactly what we're doing. We're identifying modules and then, these patterns of topologic overlap, and then asking, do they interact with each other, like across diseases or across brain regions? Are they the same? Are they overlapping? And then we can also identify their transcriptional drivers, right? By doing motif analysis, which is essentially what scenic does. And then that takes it a step further. So we can identify the motifs and their transcriptional drivers exactly in these. I hope that answers the question. But feel free to email me if it's not clear. And then the second question, is there a difference in gene expression between verbal? We don't know yet. This is why, in fact, we know almost nothing about how this gene expression correlates with any phenotype. We need many, many more people. And so this brain bank has to grow. And so my sense is it's obviously a very tough issue to collect tissue from, postmortem tissue from autism and to sign up for this kind of thing. But I guess my thought is it's if we don't grow it, we won't know. And so I think it's a really interesting question. So we have no idea how this relates to any phenotype, but I'd like to speculate that the changes, the deficit in long-range connectivity, which would impact layer 2, 3, probably leads to a change in local circuit activity that may have to do with the detail focus, not just in occipital lobe, but in other sensory regions. And so they ask, they ask, do you use WGCNA? And yes, we use WGCNA. In fact, I was kind of involved in developing it with Steve Horvath originally. We had a master's student, but then this postdoc of his, Bin Zhang, really did the major work on it. And so it's this work. I cited the paper in one of the slides, Zhang and Horvath in 2005.
So there's some. Go ahead.
A question to YouTube: what do you think is behind the phenotype expression gradient? So the gene expression gradient, we don't know, but we have a hypothesis that it has to do with the expansion of what is called like deep layer 3, layer 4 neurons that get thalamic input. And so it could really be related to the sensory processing and other sensory motor deficits that are often observed, or let's call them difficulties. And so we're beginning to do studies looking across several sensory regions, as well as their thalamic nuclei that are involved in gating them, to actually see if that's the issue. So that's a great question. That's our hypothesis. We actually don't know. But it seems that those are the most disrupted, and those are the cells that get, so the cells that are most disrupted are those that get thalamic input in the visual cortex, and that's the area that's massively expanded in humans and in primates, and as well as layer 2, 3, which are also expanded as well. So it's layer 2, 3 in the more anterior regions, which is involved in cortical-cortical connectivity, and then layers kind of 3, 4 in posterior regions, which is, getting thalamic input as well as giving cortical input. So that's our thinking.
There's another question on YouTube from Eric Cuevas.
Yes, hi. Do you have found VIP neurons or, yeah, absolutely. You can, what I didn't show you with the single cell data is that we spent 2 years trying to put all the single cell data, which is basically almost 1 million cells, into the kind of current Allen Brain or Brain Initiative atlases. And we're able to do that. So we've done that. There are some cells that aren't seen there because they're reactive or disease-related cells, obviously, states. But so we've heavily validated them. We identify every single interneuron and projection neuron class that's been identified. So yes, and there are alterations in parvalbumin neurons, a little bit in VIP, but the most profound are in the SST. But in fact, when you look in bulk tissue, in our original study, Irina Vonego, 2011, the most downregulated gene, I think at that time, was parvalbumin, or it was one of the top 5, and somatostatin. And so there definitely is dysfunction of local circuits and interneurons that's occurring. Again, how much of that is primary, there's some, and how much of that is secondary to the kind of alterations in the projection neurons is still not clear. But that can be studied now. Great question. Thank you.
Okay, so I don't see more questions. So I have some questions, but are quite general. One is, there are model organisms for the study of autism.
Yes, so people have, we and other people have knocked out genes in mice and observed behavioral changes that might parallel those observed in autism, like repetitive behaviors or problems in social interaction. It's hard to make that direct link, though, but certainly there have been some, and we've even used them for some treatment development and shown some effectiveness and even begun to identify the mechanism. And what's interesting, in one of the mouse models with a recessive gene called CNTNAP2, knocked it out. And there's several papers published, three of them from our lab on this, one in 2007 in Cell, one in 2015, Science Translational Medicine, and the most recent one in Neuron, where we show that actually the social defect is likely related. We do functional MRI and iDisco brain mapping, and we show that there's a change in network activity. There's a disconnection of regions that we can then reverse with a drug that reverses the social behavior. So the mouse models are interesting. It's hard to know how far we can take them. What I'm particularly interested in are the stem cell organoid models, which can model neurodevelopment in a dish quite well. And we've been doing quite a lot of work with our collaborator, Sergio Pasca at Stanford with that.
Mm-hmm. Okay, thank you. And the other question is, if among the genes that you observe differential expression, what happened with the stress response genes, for instance, general stress like oxidative stress or temperature stress, another kind of stress? Are they differentially expressed in the patients than autism?
Not that much. We find in the most recent cortical study, we find a module that's related to ER protein folding, but it's not a classic stress module.
Yes, no.
Not classic, it's more ubiquitination and protein metabolism, as well as splicing.
Okay. Okay, I think there's another question in the chat.
The polygenic, yes, sex bias. So somebody also asked if we're studying women. Of course, we want to study women. In fact, we have grants to study male-female differences. There just aren't enough brains out there. The ratio of male to female is 4:1. So it's just been really, really hard. But I think that's a super important area. And so we have a big grant with Simon Baron-Cohen in the UK that's funded by the Simons Foundation to do exactly that. We haven't, we have a paper, two papers, but one big one from a former grad student, Donna Whirling, in Nature Communications a few years ago, where we actually study the sex differences. And just to what we know right now is that the autism genes themselves do not show sex-biased expression. But what we see are that the genes that are differentially expressed in autism postmortem brain overlap with male upregulated genes in neurotypical males. So we think what's happening is this is really the sex differences have to do with the brain organization and differences in male and female brains that make female brains more protected and more resilient. And that's what's going on there. So that's our model. There's a paper, Whirling et al., Nature Communications, and Donna Whirling and I, when she was a grad student in the lab, wrote a few reviews on this topic as well. One in PNAS and one in, might have been in Current Opinions. So please take a look at that. That's, again, email me if you can't find them. Then the last question is, how do you calculate the polygenic score taking account? Oh, well, the polygenic score you get from huge GWAS. You need huge samples to get a polygenic score that differentiates between case and controls. So that's out of box. So that's done with the Psychiatric GWAS Consortium. And we use those data from the Grove et al. 2019 Nature Genetics paper. And then we apply that polygenic risk onto samples and things. And so what we've done is we've taken the GWAS, the heritability from Grove et al., and we've partitioned that heritability across the genome, asking, is it enriched in certain modules, or using MAGMA or LD score regression.
Okay, I think there are no more questions. So Daniel, thank you very much. It's a fantastic talk. So I think we all really enjoy it. And thank you very much. And I hope next time we'll be in person
Thank you. And thank you for all those wonderful questions. They're right on. I really appreciate it. Great questions. Great discussion. Thank you.
No, thank you. And see you soon, I hope.
Yep, me too.
Okay.
Take care. If
Take
you're
care.
in LA, look us up. Bye-bye.
Okay, bye-bye. Gracias a todos. Bye.